and welcome over to today's lecture. We are working on the fourth lecture in which we talk about the patterns of evolution and additional evidence for evolutionary theory. Got a roadmap here and we'll blow on by it. So the first one we want to talk about is the scales at which we see evolution. Right? So this has been something a lot of people talk about. Especially people trying to argue against evolution. We're going to be talking about evolution occurring at small. Oh, gotta select my pen. Evolution occurring at small scales versus large scales, and often what this will translate into is evolution occurring over short periods of time. Whereas in large scales, we're often talking about long periods of time. And when I say long, I mean long. We're talking geological time, where a human lifespan is barely a blip, right? So, when you mention evolution over short periods, you're going to be talking about evolution within a population. Remember, a population is all the members of a single species in a given area or habitat. And this is literally where we talk about the change in gene frequency, in which adaptive genes become more common over time, right? As we breed and the organisms with adaptive genes have more energy to feed and breed, their genes, as they feed and breed better, become more common in subsequent generations. And maladaptive genes become less common. And the term for this shift in gene frequency within a population is adaptation. So, microevolution is all about adaptation of populations. Macroevolution is where we talk about the big evolutionary changes, right? Speciation. So, right here, we have our little bug on a leaf, and so beetles that camouflage better with the leaf will survive and reproduce more effectively than beetles that contrast with the leaf. Selective pressure here would likely be predation, because the predators can see the contrasting beetles better, and thus the camouflage beetles survive and reproduce and pass on their genes for efficient camouflage and become more common. They are becoming adapted to their surroundings, specifically adapted to blend in with their leaves. Whereas over here, we see our dinosaurs, right? So this is large evolutionary patterns in the group Dinosauria. So this is a great example of Macroraptor. We have some old dinosaur right here, some common ancestor to dinosaurs. There were two main branches, Saurischia and Ornithischia. And then Saurischia divided into, had a common ancestor of Sauropodomorpha, and then a different lineage which branched into the large theropod dinosaurs, and Maniraptora, the bipedal, smaller raptor-like dinosaurs, and of course the avian dinosaurs. So, and that's just general trends that we can describe based on fossil evidence. So here's a great example of microevolution in the Galapagos finches. So the Galapagos finches live only on the Galapagos Islands, and it's actually, because the Galapagos Islands are so small, very easy to look at patterns of adaptation. So finches had been observed. There was an experimental study that observed finches for quite some time, and during that study, the finches on the islands experienced a drought between 1976 and 1977. There was a drought, and thus we were able to see how the birds adapted to that drought. So these are the medium ground finches. They do not have an extremely large broad beak, but it is still a broad beak, which means these finches are adapted to eating soft seeds and maybe small hard seeds. Right? The problem is, during a drought, soft seeds are basically wiped out, and small hard seeds don't survive as well. During a heavy drought, it's the larger seeds that survive the longest, and thus you have to have a very broad, thick beak to take advantage of that food source. And what we found was that there was natural variation in the finch population, beaks ranging between 8 and close to 10 millimeters in depth. And before the drought, the average beak depth was somewhere just shy of 9 millimeters. And then after the drought, the average beak depth had increased almost an entire millimeter. So by the time the drought ended, the beak depth had increased by almost a millimeter. So, and then when we look at longer term studies that chart beak depth, we can see that during dry years, we see bumps in beak depth in the population, because the birds with the broader beaks during dry years get more food and have the energy to reproduce, and thus pass on the genes for that broader beak. During wet years or average years, right, it's not like we see a plummet in the broad-beaked population. It's just that the birds with some of these beaks with smaller depths survive pretty good at just the same, and they can reproduce. And so you'll see sort of the average go down. So, pretty cool example of adaptation via natural selection in this set, with the selective pressure being water and food availability. All right, another good example would be the peppered moths. So we have peppered moths in North America and the British Isles. British Isles are smaller and the habitats are smaller, so we have much more data on them. So the big things about these moths is that they have an adaptation for cryptic coloration, and that is our fancy science term for camouflage. So they are camouflaged from predators by blending in on the tree trunks that they rest on. There tend to be two morphs of these peppered moths, right? There's a melanistic or dark morph, and then the standard white and black cryptic morph. Right? This is cryptic for these ash trees, or birch trees, I think, that they blend in with, and especially the lichen that tends to cover those trees. So, the predator, the selective agent, is a bird. And basically the bird sees melanistic moths much more easily and takes them out, eats them, such that it's the cryptic moths that are able to survive and reproduce. However, during the Industrial Revolution, right, we had massive increases in pollution due to coal. Wow, thank you. Stupid PowerPoint. Coal-fired factories. So you had this proliferation of factories that are using coal power, and that belched out massive amounts of smog, and the smog ended up staining the lichen, and the lichen became blackish brown. And suddenly, our cryptic moths weren't cryptic anymore, whereas the melanistic moths were suddenly cryptic. So if you're a bird looking for a meal, right, you got two options. It could be this melanistic moth or this cryptic moth, and what do you know, that poor cryptic moth ain't so cryptic anymore. And during the years following the Industrial Revolution, the late 1800s, the dark moths started becoming significantly more common. Their genes became more common in the populations of peppered moths. Now, the good news is that after about the 1970s, we saw some important pollution controls. So these pollution controls, where's my pin? There we go. Oh, come back here. Give me my pin back, computer. These pollution controls served to drastically reduce air pollution. And when you reduced air pollution, you reduced smog. And when you reduced smog, your new lichen returned to its original, or were at their original colors. So, there we go. So we can see in 1959, as late as 1959, we still had a significantly high number of melanistic moths. By the way, blue dots are for British Isle populations. Red dots are for North American populations around Michigan. Anyway, you can see we have much better info for the British ones, but you're going to see a similar trend. So in the mid to late 70s, when the EPA was established by Nixon, we had a massive improvement in pollution, and now we're starting to see the original pre-industrial revolution rates of melanism in the population. So again, selection favored the cryptic moths, and the melanistic moths were once again maladaptive. So, adaptation. All right, now on to macroevolution. By the way, macro and microevolution are not distinctly different from each other. If you have microevolution, you have macroevolution. There is no fundamental difference between them. It is just sort of the scale and often the time it takes to see this. So, there you go. In macroevolutionary trends, what we're going to be looking at is the trends over time. So, for instance, long-term trends, we see three different kinds of mammals. Eutherians, the placental mammals, of which we belong. Then there's the marsupial mammals and the monotremes. Monotremes are the egg-laying mammals, including our duck-billed platypus, which is a pretty wacky animal. It was not believed to be a real animal when naturalists first described it. It was because at the time the Royal University in England, where all these naturalists were reporting to, had recently been rocked by a scandal in which a naturalist off the coast of South America had been describing species after new amazing species and then found out as having been a creative taxidermist, in which he had been sewing different species together to make new animals to describe so he could be super famous. So they'd been rocked by that scandal, and then along comes a guy who describes a small furry mammal that has a beaver tail, an otter body, lays eggs, has venomous spurs on its ankles, and a duck bill. And they say, yeah, no, go away. And then he brings a taxidermied one back, and they say, ha ha ha, good taxidermy skills. And it wasn't until he finally brought back a live platypus that the Royal Society has actually accepted the platypus as a real thing, which is kind of funny. Anyway, we can look at the evolutionary trends in Mammalia from the original common ancestor, the ancestor to all mammals, somewhere around 175 million years ago, and the divergence. So here's the lineage diverting to monotremes versus the other mammals. And then here's the common ancestor, and we have the diversion to the marsupials and the eutherians. Eutherians becoming significantly more common, with marsupials basically being reduced primarily to Australia and Tasmania. There are marsupials throughout the rest of the world. The opossum is a marsupial. So, and then of course there's a little marsupial species in South America representing the oldest species, the oldest extant species of marsupial in the world. Extant means still alive, basically. So a species can be extant or extinct, and it could also be extirpated. If it's extirpated, it's been driven to extinction in a specific area. So the species is not extinct, but they're all gone from a certain part of their native range. So wolves have been extirpated from significant amounts of the United States. Bison have been extirpated from most of their range. So anyway, yeah, the oldest extant marsupial, the one representing the most ancestral marsupials, lives in South America, where their evolution started. Now, we see this trend. So how do we know common ancestors of mammals appeared around 175 million years ago? How do we know where these divergences occurred? And how do we reveal patterns of macroevolution? Well, it is, as you might have guessed by now, the fossil record. The more fossils you have, the more patterns you can generate. And we have significant amounts of dinosaur fossils. So our fossil record for dinosaurs is beautifully complete. So here we can just see significant patterns of evolution in the dinosaurs. And here's our avian dinosaurs right there. So that's pretty cool. Here we see another great example of of the fossil record, showing patterns of evolution with our little key events, right? Bipedal walking, right? Enlarged cheek and jaws, a larger brain, tool use, right? And even larger brain. So we can see all these different species of apes. Here's our extant great apes, chimpanzees, and bonobos that are most closely related to humans. And then when we have our bipedal humans, we get into a pretty cool group called hominids. Bipedal walking apes. So the hominids are or primates, I guess I should say. Ape specifically refers to chimpanzees and bonobos. So we're bipedal walking primates. Got to be technically correct, because in science, if you ain't technically correct, you're incorrect. So here we go. These are the different hominids we can see in the fossil record, and based on important features of the hominids, we can differentiate from different species, and based on when we find them in the fossil record, we can develop a tree showing common ancestry, relatedness, and all that nice stuff. And it gives us this beautiful pattern of evolution of the hominids. So the pace of evolution can be described in two ways. When Darwin first described evolution, he specifically and only described the time over which evolution occurs as graduated. In gradualism, we have very slow changes over long periods of time. So small changes over long periods of time. And gradualism is a great illustration of how a lot of speciation has occurred. So patterns of speciation can be illustrated through gradualism. However, we also have punctuated equilibrium. In punctuated equilibrium, what we see is long periods of basically stasis. So long periods of little change. Indicating that over these long periods, our species are well adapted. And then, quote, short periods of large amounts of change, indicating that we have new adaptation occurring. So we need to adapt to some new condition. So, here we have a good example of punctuated equilibrium. This is the shell of a fossil foraminiferan, which is kind of an interesting little snail. These are recent fossils. Older in the fossil record, they were smaller and sometimes a bit rounder. And what we can see over millions of years, from the Miocene to the Pleistocene, we see for roughly, 6.5 million years, shell size remains and shape remains pretty stable. So then suddenly, by the time we get to around 5 million years, shell size has changed. So shell size and shape. We've gotten larger shells, and they've moved away from the rounder shell and more into this sort of large conical shell. And it was just that brief period, just this brief 500,000 years, and then we saw this big change, right? So this, by the way, is what we mean when we say short periods of rapid evolutionary change. So in stasis, you see, here's our fossil record, and we see our conical, our shells, and all the species are still showing that shell. And then when you have our punctuated equilibrium here, we can see a rapid shift in shell shape. Often requires isolation from a larger population, and some change in the environment that this isolated population must adapt to. So, anyway, pretty cool stuff. Punctuated equilibrium. And we've got another example of punctuated equilibrium in the wild sunflowers. This is a naturally occurring species, Helioanthus anomalus. Right? So basically, it suddenly appeared and speciated in the fossil record in a hybrid zone. So it basically just suddenly appeared and speciated, and it did this in the hybrid zone for two other wild species of Helioanthus. And scientists were trying to figure out, how long it might take for this species to appear. And they ran an experiment to examine the sort of genetic changes required for this hybrid to speciate into the modern wild sunflower. And so they took two species, the two different species that meet in that hybrid zone, Helioanthus petiolaris and Anis. And they artificially, they pollinated them and created a hybrid generation. So here's the original hybrid generation. And it was quadruploid. It had double the number of the parental chromosomes. And it had roughly 10% fertility rates. It wasn't all that great at sustaining itself. However, as they went through multiple generations in their experiment, artificially pollinating them, they saw two major chromosomal fusions in their experimental hybrids. Right here in chromosome 1, they saw a fusion of two chromosomes into one, and here in chromosome 2, they saw another fusion of two chromosomes into one. And these fusions popped fertility up to 90%. So these simple fusions drastically increased fertility. And in fact, when you sequence the wild Helioanthus anomalus, you can see two chromosomes with evidence of fusion. So it's likely that in the hybrid zone you had these precursors to Helioanthus and anomalus reasonably occurring over time, just through random chance, and then 10% of the time they were able to breed, and then over time, at some point, a fusion event took place in one or more individuals, and suddenly you were able to breed, and the species rapidly became distinct and self-replicate and able to breed and create a new population. So that's a pretty cool example of macroevolution in a short period of time. At least we can experimentally produce it in a very short period of time. Okay, so we talked about how fossils are our evidence for evolution, for macroevolution. Fossils show us the pattern. So the first thing to note about fossils is that fossils are very rare. In the fossil record, fossils are very rare. This is because there are some very special circumstances required for fossilization, right? We have to fossilize before the fossils decay. So we have to prevent decay in some way. So we have to prevent decay to prevent all the tissue from breaking down, including the bones. Bones will break down and decay into much more brittle forms. We need to fossilize before those brittle bones are pulverized. And in addition, we need to prevent scavengers from getting a hold of our corpses. Because scavengers will break apart the body, scatter the bones, and many scavengers will crack bones to pieces to get to the marrow inside. So we both have to prevent decay, and we also have to prevent scavengers. And what this generally means is that an organism, in order to become fossilized, must be rapidly covered in sediment. Rapidly made unavailable for scavengers, rapidly covered up in something that will prevent oxygen from reaching the bacteria that cause decay. So, whether this is from a flood, right, volcanic ash, tar pits, they need to be covered in something that limits oxygen availability to prevent decay, limits weather exposure to prevent decay, and limits general exposure to prevent scavenging. So flood, volcanic ash, tar pits, and other things like that. So we have to rapidly cover in sediment, and we have to prevent decay and scavenging. So as you can imagine, this is not a common event, right? Roughly, I don't know, we think that roughly 99 plus percent of organisms are not in the fossil record. Right? Because it's so rare, and in addition, because the fossil record favors certain species being fossilized, right? It favors hard structures, hard mineral structures. Right, so this would be things like exoskeletons. We have a lot of insects out there, shells, tons of fossil shells, and of course, bones. So this pretty much eliminates almost most soft-bodied organisms from being in the fossil record. It also favors larger organisms. If your bones are very small, the geological processes associated with fossilization may pulverize them. And then lastly, you're more likely to find fossils for the most abundant organisms. What that means is that you are going to limit your ability to find organisms with small populations in the fossil record. You're going to eliminate your ability to find organisms that are adapted to microhabitat. Basically, they're highly adapted to only a certain area, because again, if you're adapted to a microhabitat, you are less numerous, right, and harder to find. So it's like you're likely to find fossils that meet the favored conditions that are of species that were the most abundant in their era. So they were abundant, they were on the planet for a long amount of time, and there were enough of them that The randomness of the universe allowed them to be in those specific circumstances that allow for fossilization. So, original dating of fossils back in the fossil craze of the 1800s was done through relative dating, in which we basically dated fossils based on the layers that we find them in. So, when you look at rock, it's generally stratified into multiple layers. So, this is a good example of simple stratification. And we have 1, 2, 3, 4, 5 layers of rock. And if you found a fossil here in the dark layer, you'd be like, oh, well, it's 2 layers down from the present, so it's, however old you might assume that is. And it even works well in hilly terrain, but geological processes can produce folding. So if you have some event that causes a little bit of uplift here, and the crust moves just a little, right? We don't have any problems still doing relative dating. But if we then have a shear force that breaks that and then causes it to roll, we can end up having these layers where it's still those 5 layers of rock, but when you observe it, it's 1, 2, 3, a little 4th one here, a 5th one, a 6th one, a 7th, and an 8th, and even a 9th. So if you find this fossil here, right, and over here, it will appear to have been around in the fossil record much longer. So relative dating is nice because deeper layers mean older fossils, but it's error prone. So it is prone to errors. So relative dating is where you compare the position of the fossils to the layers around them. And it is error prone due to things like overturn layers, right? You can have overturned layers. You can have uplift events which change how the layers look. There's lots of stuff that can cause changes to the appearance of the layers and mess with your ability to do relative dating. Relative dating works great where you have these nice, beautiful, regular stratifications. So, for instance, here's a nice, beautiful canyon, and you can see there's no overturned rocks. There's no major uplift events, just nice flat layers for comparison. So it's very easy to do relative dating in places like the Grand Canyon. But if you want to get rid of some of those errors that you often see in relative dating, you should do radiometric dating. So radiometric dating gives you the absolute age of a rock. So it gives you the absolute age of rock layers. Right? I put absolute in quotes because there's always a margin of error. So it might be as small as plus or minus 8%. So you've got a 92% accuracy in the age of the rock. But if that age is something like, 800 million years, 8% is like 100,000 years. So, it would be 800 million plus or minus 100,000 years. So it's not like, this rock is 50,362,252.8 days or years old and was made on a Tuesday. Yeah, you don't get that from radiometric dating, but you get a pretty darn accurate numeric age of that rock. It's based on radioactive isotopes. If I had to ask you for examples of radioactive isotopes, probably the most common one people talk about is carbon-14. Carbon-14 is a radioactive isotope. What does it mean to be a radioactive isotope? Well, it means that you have an uncommon number of neutrons in the atom's nucleus. In this case, the normal number of neutrons in a carbon nucleus is around 12, and carbon-14 has 14 neutrons, and thus, it's 6. Anyway, and it has 8 neutrons. Anyway, the important thing is that it's a radioactive isotope, which means it has an unstable nucleus. An unstable nucleus emits particles over time. It emits energy and particles. That gamma radiation coming off of things like radioactive uranium ain't very friendly. The particles it emits includes protons. And when you lose a proton, you change the element that you are. So carbon-14, being a radioactive element, goes through a decay pathway. Right? So all radioactive isotopes have a decay pathway in which they decay from one to another element. It's usually a chain of different elements until you find the element that you decay into that has a stable nucleus. And then you stop kicking off protons and energy, and you are now a stable element. What this means another amazing thing about the decay pathway is that each step in the pathway always occur at a constant rate. Right? So you know the rate at which the elements decay from one step to another. And thus we are given the concept of the half-life. A half-life is the time required for half of the radioactive element to decay to whatever step you're looking at. The next step, or maybe even you're just going final step. And because the half-life, because you're looking at radioactive decay, and those steps occur at a constant rate, the half-life is a set amount of time. And so simply by looking at the ratio, of the radioactive isotope compared to the ratio of the amount of radioactive isotope to the next step or final element, whatever you're measuring at, gives you the amount of time. How does that work? Well, for instance, carbon-14 has a half-life of somewhere, I think, around 5,000 years. Carbon-14 is great for dating recent fossils because there's often intact bones in recent fossils, like Ice Age fossils. And carbon-14 forms in the atmosphere And as part of normal processes, whenever you breathe oxygen in the atmosphere, you're also breathing everything else in the atmosphere, and you'll get carbon dioxide into your body. Plants take in carbon dioxide. Some of that carbon dioxide is made with carbon 14. And because there's generally a specific percentage of carbon 14 that forms in the atmosphere, just due to random chance, stays pretty much at a steady rate, you incorporate a specific percentage of carbon 14 into your body. And then all you got to do is look at the ratio of carbon 14 in an organism compared to the final step in the decay pathway, and then you know how many half-lives you've got, and you can compare that to the amount of time required for each half-life. So here's the uranium-235 decay pathway. Right, so uranium-235 is a radioactive isotope. We use it in dating. It's also used in atomic bombs, and also nuclear power. So uranium, thorium, proactinium, a different isotope of uranium, then a different thorium again, then radium, then we have astatine over here, polonium over here, bismuth, lead-214, another polonium, a titanium, lead-210, bismuth-210, polonium-210, lead-206, titanium-206, mercury. Anyway, this lead here, I hit it off earlier, but this lead is our final element, and it has the stable pathway. So uranium-lead-carbon dating is reliable for 1 to 2 million years. It has billion years. So it has a reliable. Sorry, I read that wrong. It's reliable for any date range between 2 million to 2.5 billion years. Give me my decimal back. So from 2 million to 2.5 billion years. This is the date range for which uranium dating is reliable. And this is uranium-lead dating. So we're talking about the half-life to go all the way from uranium down to lead. People often do uranium-thorium dating, which is literally just this first step. So anyway, the half-life is around 700 million years. So it takes 700 million years to go from 100% U-235 to 50% U-235, 50% lead. So if you have, 25% of U-235 left in the specimen and 75% lead-206, then you went through a half-life to get to 50%, divide that in 2, and another half-life to get to 25%. So that's 2 half-lives, which is 700 million times 2, which gives you 1.4 billion years. Right, so this is some pretty easy math as far as calculating, as far as calculating how long it's taken for that dating for that decay to occur. Just look at the ratio, right? Just look at how much is left. Right here we have the 100% of the original amount is left. Then you have a half-life and you get to 50%. Divide that in half again and you get to 25%. Divide that in half again and you get to 12.5%. Divide that in half again, and you get 6.25%, right? Or fraction-wise, 1/2, quarter, eighth, 16th. And you can count how many half-lives have occurred. So to get to the 50%, 1 half-life has occurred. At 25%, 2 half-lives. 12.5, 3 half-lives. 6.25, 4 half-lives. If the half-life is 5,000 years, how old is the fossil if you only have 1/16th of it left? Well, that's 4 half-lives, 5,000 years. Your fossil is 20,000 years old. If it's just 2 half-lives and you've got 25% left, it's 10,000 years. So the math on this isn't hard. And it's super reliable. We can use, for instance, radioactive potassium to argon dating for the oldest volcanic rocks. Uranium thorium is good for a couple 100,000 years. So it's good for the ice ages. Carbon-14 is 60,000 years out. So that's pretty good for recent fossil history. So, again, it's important to note that you don't date the bones. You don't date the fossil bones themselves. You date the rock layer that the fossils are found in. So we're radiometric dating rock layers, because often there's no bones left. Okay. Evidence of evolutionary change in the fossil record is often presented over geologic time, which is time frames that are much larger than human brains really handle. You're talking about, periods of 10,000 years, 50,000 years, 200,000 years, a million years, a billion years. This is geologic time. Geologic time is very different, right? The fossil record is good for examining changes and patterns over geologic time. Give me my A back. So there's a lot of great ways to see these patterns over geological time. One would be just looking at intermediate species. So fossil intermediates. A fossil intermediate is a species that shares key characteristics, but has some sort of body form or some kind of form physiologically shows a change in form between two species. So say you find a fossilized bird with claws and teeth, and then you find a fossilized bird with much smaller claws, and its fingers are shorter outside of the wings, and it doesn't have teeth, and then you find a fossilized bird that looks very much like a modern bird. The one with the shorter claws and fingers that barely extend out of the wings, and the one and having no teeth would represent an intermediate form. It still has features of the previous fossil, the claws, but it has features of the modern fossil as well. So it's showing a change in form over evolutionary time. So this is a species from a lineage that has occurred later in evolutionary time. Because it shares key characteristics between them. They're in the same evolutionary lineage. Because it's showing that intermediate form, it's giving us that pattern of claws disappearing and teeth disappearing in order to produce modern birds. So, we expect gaps in the fossil record. Gaps are expected because remember, fossilization is rare. A gap, by the way, when we talk about a gap, what we are talking about is a lack of species showing that intermediate. So we expect a lack of intermediates given that fossilization is extremely rare. However, even then, when we look at fossils for, say, our extant rhea bird, where is it? Yeah. Here's our extant rhea, which we found in fossils as far back as like a couple million years. So rhea's been around. When we look at fossils showing similar characteristics, right, we find an awful lot of them. So we don't have an intermediate for literally every single step, but we have an awful lot of fossils showing an awful lot of intermediate characteristics. So even though we expect gaps in the fossil record, we have a significant amount of known intermediates. This is just a scale of hominids, right? Here for reference, we have a chimpanzee skull. So this is the extant great ape to which we are closely related. And then from here on out, we have skulls of various hominids, each one representing a different species. All right? So this final one is our only Homo sapiens skull, modern humans. This second one, I think, is Homo neanderthalensis. So Neanderthals. This one is probably one of the other ones. Could be Heidelbergensis. This is probably Homo erectus. We've probably got an Australopithecus or two down here. So we have a significant number of known intermediates, and we see these very similar characteristics, which gives us the information that these are indeed closely related species. All right, moving forward, here's Archaeopteryx, another great intermediate. Archaeopteryx had a lizard-like skull. Well, not lizard-like. It had a more raptor-like skull. It didn't really have a beak. What I'm trying to get at is it didn't really have a beak. It had teeth. It had claws, but it also had large wings, and its tail, though long, was highly feathered with complex large feathers, like the kind you see in bird tails. So large wings like birds. Long or a highly feathered tail like a bird. However, its tail was long, like the raptor dinosaurs. It had claws similar to its raptor cousins, and it had teeth and skull similar to them. So this represents an excellent intermediate between raptor dinosaurs, the Maniraptor dinosaurs, and the avian dinosaurs. Now, an intermediate is not a direct ancestor. It is not a direct ancestor. It could be, but it does not guarantee that it's a direct ancestor, right? So this might be the lineage that you get to for avian dinosaurs. And you have various splits showing various different lineages in that evolutionary history. And then this one over here might be where you get your Archaeopteryx. So you might see that it is not obviously a direct ancestor. But it is a good intermediate, showing the evolutionary pattern that would produce avian dinosaurs. Here's a great example for the evolution of modern whales. And the evidence is that they evolved from land mammals, specifically this land quadruped. Here's Pakicetus atakai. It lived on land, but its skull had very distinct characteristics shared with modern whales and dolphins. It's generally associated with like tooth shape, nasal bones, nasal cavity, some very distinct characteristics that are shared only amongst modern whales. And so we look for those key characteristics. Here's Ambulocetus natans, and you can see we've got a reduction in limbs. The forelimbs have gotten smaller, likely more flipper shaped. The hind limbs are still long, so it indicates that it still goes onto land at some point. Then we have Rhodocetus kazarani, and Rhodocetus here has an elongated body and tail. We have evidence for flukes in them for swimming, that horizontal shaped fluke. Their hind limbs are reduced to the point of being functionless, and they also have that paddle fin in the front. And then here's modern toothed whales. So by simply seeing the shared skull features that identify it as a key ancestor of whales, we can chart a very cool evolutionary history. All right, continuing forward, we have the evolution of horses. And what we can do is some paleoclimatology, looking at the types of plants that were present in the environment. So we can get some climatological data. The earliest ancestors of horses, way down here, Hyracotherium, right, were very small. And in the fossil layers we find them in, we often find fossils of jungle plants. So they browsed amongst the leaves of jungle plants. Over here we have Myohippus. It's a little bit larger, still a grazer. Right? If you continue through, you see a larger horse over here. This is not a direct line ancestor of modern horses, but it still shows all the characteristics to be in Equus. So over here we have a differentiation. We can look at the teeth and the plants that they're found in and find that there was a proliferation of grasslands. So during horse evolution, we had a shift in climate, and thus a shift in their ecological area, their geographic area, to become more full of grasslands. And that increase in grasslands meant a evolutionary adaptation towards a life as a grazer. Now, grasslands also have much less dense foliage, fewer trees, so we also see a pattern of these horse species getting larger, till you have modern horses that are extremely large compared to their ancestors, well adapted for grasslands, for running. So our major changes are a size change, In addition, we have a change in toes when you're moving around in the jungle in the uneven terrain. Having multiple toes is nice for gripping onto stuff. Right over here, we have 3 toes. As you have a transition to the grasslands, you see a reduction in those spare toes and an adaptation towards running, the development of hooves. So, you end up with 4 of the toes being elongated to become a large middle toe with a hoof for running. You can only run real fast on grasslands, not through a jungle. And then your teeth, they get larger and tougher for chewing grass. So these are great examples of patterns of evolution in horses. Again, evolution is not goal-oriented, right? Evolution is not goal-oriented. You have the constraints. Evolutionary change is based on change in existing structures. Evolution acts on existing traits. It cannot magically crank out a new trait based on what is needed. So, evolution of giraffes is based on the constraints of their mammal ancestors, and what that means is that giraffes, just like humans, only have 7 vertebrae in their cervical, 7 neck vertebrae. So, cervical, which is neck vertebra. Giraffes have an extremely long neck. Humans have 7 neck vertebra. Right? So we didn't magically crank out a bajillion new vertebrae to get a longer neck in a giraffe. What you see is an evolutionary change on the length of the vertebra, not new vertebra. So evolution did not set out to elongate the neck. It did not say, well, I have a small-necked ancestor here. Why don't I crank out a long-necked ancestor? So we'll just add about 30 more cervical vertebrae. Boom, long neck. So geese have 25 neck vertebrae because they have similar numbers of vertebrae to the other avian dinosaurs around them. So geese have extremely long, very flexible necks. Giraffe necks are comparatively inflexible. They actually have to spread their forelegs out wide in order to actually lower their head down. Giraffes cannot easily bend their neck. Their neck has less movement in it than our neck. See, we can bend our neck pretty easily because our vertebra, our cervical vertebra, are short and there's very few spaces, so it's relatively easy to get a good bend in there. With the giraffe vertebra being so long, because there's only 7 of them, they can't bend their heads really. Most of the flexion they get when trying to reach the ground with their heads comes from the base of the neck. Right? They've elongated, they've become thicker, stronger, supporting much more musculature and tendons to keep the head up at the top of the neck and keep the neck straight. So that's a constraint based on the existing structures. Evolution is based on ancestral features. Again, we can look at the eyeball. The eye is a beautiful example. If we compare the eye in vertebrates, right here, versus the eye in cephalopods, right, like squid and octopus, the eyes share similar features. There's a lens. There's musculature supporting the lens. The lens can be flexed to get a little longer or shorter and fatter in order to focus on objects at different distances more easily. And they have light sensing cells along the back layer called the retina. And light comes in through the cornea, bends towards the retina, focused by the lens, and bam, you get sensing of light. Right? Some very simple similarities. However, the vertebrate eye is based on the old constraints of being a vertebrate. And what that means is that while the light sensing cells are here, this is my, a little box. This is my light sensing cell. A cell that emits a signal when light hits it, right? It is connected to a nerve cell, which will send that information to the brain, and then the nerve cell is connected, has its nerves connected here, and this is all arranged so that light coming in has to pass by the nervous layer in order to get to the actual light sensing cell. So if you need to connect up all the nerves of your light sensors in order to send all that information out to the brain, you need an optic nerve, and this nervous layer being on top of the light sensing layer means that where you have the optic nerve, you have a blind spot. Can't have any light sensing cells there because the light sensing cells are back behind the nerve. So, this is the vertebrate eye. Compared to the cephalopod eye, all the light sensing cells are right up front, and then they connect back. So here's the light sensing cells, then they connect back to the nerves, and thus the optic nerve goes right back. So there's no blind spot. In cephalopod eyes, because the nervous layer is behind the light sensors. Whereas here, the nervous layer in the vertebrate eye is on top, in front of the light sensing layer. So we get these imperfect structures due to the constraints of our ancestors. It indicates that the cephalopod and the cephalopod eye and the vertebrate eye are analogous structures. They have similar features, perform similar roles, but do not indicate common ancestry. So these developed to fill the same role, convergence. So there was convergent evolution, but one is arguably a much better design than the other. So, if you were thinking maybe the best designed is if you want a designer, and the designer wants to make something nice, why would the designer make the octopus and squid better? Is it because that's what's actually favored? Anyway, jokes aside. Alright, vestigial structures are also evidence of evolution. So, vestigial structures appear to serve, they apparently serve no function. There does not appear to be any obvious purpose for a vestigial structure. However, they are similar. Vestigial structures have a similarity to structures in ancestors or closely related animals. Closely, again, we're talking evolution, so slap that in quotes. That serve a function. Right? So, they apparently serve no function in one organism, but they are similar in structures in closely related organisms or ancestors that do serve a function, which indicates that over evolutionary time, that function was lost. Which also implies that that function was unneeded. So there we go. Whales and dolphins have a rudimentary pelvis fused with a rudimentary femur. Right? You can find them inside the body, disconnected from the spine. They serve zero function. You could argue that there's a small amount of muscle anchoring for the tail, but they basically serve no function for the whale. So why is that vestigial structure there? Well, if you recall, once upon a time, when we were talking about similarity of skull features, right, this is your ancestral, this ancestor to whales, the Pakicetus atakai, and it had hind legs because it was a quadruped. So this is evidence of their evolutionary lineage. It shows ancestry with a quadrupedal land-dwelling animal. So vestigial structures are also evidence of evolutionary lineage, as well as indicating again that evolution is not goal-oriented. Why evolve a whale or a dolphin from something that's quadrupedal and lives on land? So, not goal-oriented. It's based on an adaptation to the environment around you. So, if you look at the appendix, it is, in humans, serves little to no function, right? But it is similar to something called a cecum, right? A cecum is involved in cellulose digestion. Our appendix is not involved in cellulose digestion. And in any organism that's biased towards a carnivorous diet, like the opossum, you have a highly reduced cecum that is similar to an appendix. Whereas any animal that has a diet heavy in plants, like, say, the kangaroo or the koala, which eats extremely worthless plants that have to undergo significant amounts of digestion, zebras, they have larger, longer, more complicated cecums, serving a great function for cellulose digestion. We have little cellulose in our diet, but we evolved from an ancestor with a cecum, so we have evolutionary leftovers. The appendix is a vestigial cecum. Almost done. Extinctions are a part of evolution. Most people think of extinctions as bad, right? But extinctions are a natural part of life. Occasionally you have a large shift in conditions in some areas, some large areas, or even global conditions, and you have a mass extinction, right? Here, extinction rate peaks, right? The blue is the average extinction rate. Here's my pen. And here we have a massive spike in extinction rate. If we look at it, it's actually associated with The appearance of photosynthetic organisms, which increase the amount of oxygen in the air and water. So, life before that had evolved and adapted to an atmosphere with little oxygen in it. You go back in time before this point, you're probably going to have extremely limited energy because there won't be enough oxygen to support you. Might even suffocate to death. So, this mass extinction was due to conditions required for modern life. Then you have this massive extinction here between the Paleozoic and the Mesozoic, just before the Triassic. Here's another mass extinction right at the end of the Cretaceous due to the KT event, that big old meteor that blew up a large amount of the Yucatan. What the heck? Oh, that's my phone alarm. I have to be somewhere soon.
Learn more at eahimmigration.com.
So, major extinctions. We have the Permian extinction 250 million years ago. 96% of all plant and animal species went extinct. There were massive volcanoes, like these huge volcanic plains with massive numbers of massive volcanoes. And it appears that there were huge eruptions during that time period, enough to put enough ash in the air to 96% of all species went extinct, right? In each of the 5 mass extinctions that we see, the 5 peaks, here's I've circled the biggest, but we've got another one here, and another one here, and here, right? So 1, 2, 3, 4, 5. So anyway, 50% of the marine species have died in each of the 5 mass extinctions, which has drastically changed the look of the species in our oceans. We don't have any aquatic reptiles that you saw, like the mosasaurs and the plesiosaurs during the age of dinosaurs, the Mesozoic now, because they all went extinct in the Cretaceous extinction. And so you had those volcanic eruptions in the Permian that just killed 96% of all plants and animals. The Cretaceous period, again, 50% of marine species and a significant number of terrestrial animals went completely extinct, including all of the non-avian dinosaurs. So that's pretty amazing. But that's allowed the proliferation of species we see today. Mammals during the dinosaur period were not extremely competitive with their dinosaur relatives. So they had they were low in number, and they occupied small roles. They were generally small mammals, often nocturnal to avoid the larger diurnal dinosaur predators, eating, plants and insects. But when the dinosaurs died out, they left all these niches available, and you had massive amounts of adaptive radiation to fill those ecological niches. So that's pretty cool. Some people are calling the era we live in now the Anthropocene. Anthropocene. Because humans are having such a major impact on the environment. Like 25% of all species may end up extinct in the near future. And the rebound is going to be slower. Like, we're overfishing. We're wiping out fish stocks due to the commercial fishing industry. We're poaching organisms that are in populations that may not be able to take it. We're polluting environments, often making them inhospitable, right? And then, of course, there's climate change due to global warming. And so all of these things combined together are cranking out a mass extinction. However, the big problem here is that all these are occurring at a faster rate than anything before, right? The Cretaceous extinction may have caused a drastic reduction in the amount of sunlight available and all this stuff. But again, that was an extinction over geological time. We start the Cenozoic, the period of mammals, at 65.5 million years, meaning that extinction occurred over something like 500,000 years. 500,000 years. Climate change has been occurring at a rate of, we measure climate change over the past 200 years. So this is occurring at a rate significantly faster. So there's even question as to whether or not species will rebound. So, hooray! Ending on a up point. Let me compensate for ending on a downer here. Give me a second. Wait for your reward, it's coming. It's a cat. Kitty, kitty, kitty. Kitty, kitty. Oh, it's a kitty, 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 kitty. Hi, kitty. Okay, go away, kitty. He wants to eat.